Hello. Yes.
Yes, ma'am.
Are you people there?
Yes,
Hello,
ma'am.
Yeah. May I know who's this?
Preeti.
Louder, please. Preeti. Shweta. Oh, that is from Krishna. Shweta. And Seema. Okay. Can you able to hear me?
Yes,
Able to
ma'am.
hear me?
Yes, ma'am.
Stop the other TV sounds or anything else. Okay. So good afternoon, one and all. So now we will have the presentation on the cultivation of crude drugs. So when you see the PharmD students, it is coming under your second chapter, and this particular part is left out. And also for the fourth sim, it is also in the second chapter, right? So now we will go on to the presentation. So when you just go to the cultivation, what do you mean by the cultivation? That is, in your houses also you will be growing up the plants in your garden, or sometimes in the open grounds or in the pots you will be doing it. So why you are doing it? You are just developing a particular plant which may be ornamental or otherwise which may be beneficial to you in your day-to-day life, and you will provide all kinds of environmental situations which is favorable for the growth of the plants. So by cultivation, you are producing the improved quality of the plants. And what does this cultivation also does? It helps in selecting the species and varieties of hybrids. And when you just cultivate the plants, you will get whatever the phytoconstituents or the active ingredients which are required for you in a controlled environmental growth without its stopping its supply. And also this cultivation makes the collection and processing steps easier when it is obtained from the wild sources. And when you just see at the end the results obtaining from the plant, that is, what is the net result? The production of the secondary metabolites, right? So the production of secondary metabolites also will be maximum at the end of your cultivation. And because you do the cultivation of the plants rather than collecting it from the wild source, it also leads to industrialization. That means if you want to supply the drug regularly throughout the year, naturally you need to do the cultivation in a very systematic way with all kinds of requirements. So naturally it's going to produce the industrialization, especially when you just see the tea estates in Kerala or in some other hilly regions of Tamil Nadu or wherever it is, wherever there is a tea industry where the skilled people are there to collect the plants as well, and also for drying and other things, and that's make the that particular place to be industrialized, and more amount of employment is provided in that particular area. Okay. And next, if you want to do any research work you in these cultivation methods, that also is possible. That means it acts as a useful tool for the research purposes. And next we will see about the advantages of cultivation. So what are the advantages of cultivation from the wild source? First thing, it ensures the quality and purity of the medicinal plants. Yes. That is, when you just go to the collection of the same drug from the wild source, in some of the conditions, what happens due to the climatic changes or other drastic changes which is happening in the surroundings, the yield of what you're getting from the wild source may be less, or sometimes even get the yield, and there will be what happens, the amount of production in the secondary metabolites may be less. So that is why when you do the cultivation, what you're doing there, you are providing all environmental condition in a uniform way, so that what happens, the yield is also more, and the amount of active ingredients present in the drug is also more. Okay. And next one, when you just see the cultivation, yes, when you just see the cultivation of rhizomes, usually when you are doing the cultivation, it requires the more amount or the more supply of your fertilizers, as well as you have to do the irrigation properly. Irrigation is nothing but supplying of water. Okay. And also the systemic cultivation, it will result in raising the crop with maximum amount of your active ingredients. So here you can see, as I already quoted the example in the cultivation of rhizome, when you are doing the cultivation, you are going to provide the required fertilizers and required irrigation, so that the volatile oil component or sometimes the resins which is present in the rhizomes will be have present in the more amount. That is example of rhizomes, maybe ginger or turmeric or licorice. You know very well about ginger, turmeric, and all those things.
Licorice.
Yes. And apart from that, if the cultivated plants, usually we will be keeping the plants when you are cultivating free from weeds. So when you keep the plants apart from the weeds, the contamination of crude drug is avoided. Okay. And next, if you see the second major advantage in the cultivation of crude drugs, this plants, that is the cultivated plants, will give you the better yield and high therapeutic quality. Yes, because in most of the cultivations, you required the skilled operations and also the professionals who are well trained in that particular area. Especially when I was talking to you about the cultivation of the tea plants or tea plantations, definitely what happens during the harvesting or during the processing, the people should be having a well versed knowledge in these things. Okay. And also there are some of the products, already we have seen the detailed method of cultivation collection of opium, right? The latex collection requires the skilled uh, people for the making the incision, as well as for the collection of the your latex. Okay. And so that is why when you just do the cultivation of the plants, usually it gives the bigger yield, as well as the compounds which are present in that will have the high therapeutic quality. So here, under this, some of the examples, as I already quoted, it is given over there. That is the collection of latex from the poppy capsule, and collection of folio resin from Pinus species, and also the preservation of green coloration during the drying of senna. So this also we have seen when we have seen about the cultivation collection of senna, that is the PharmD students. And also the deterioration of cardiac glycosides can be minimized when you are collecting the digitalis leaves and then it is dried. Usually when we are storing the digitalis, the moisture content should be less than 5%, right? So if the moisture content is more than 5%, what does it do? It initiates the action of the enzyme digiperpuridase, which is present in the drug, and that will lead to the chemical decomposition of cardiac glycosides in digitalis. Okay, so that is why when you have the skilled people, and naturally the cultivation collection is made easier. And next one is this cultivation ensures regular supply of a crude drug. So during the cultivation, how you can give the crude drugs in a regular supply, or continuously you can supply the drug. The cultivation is a method of crop planning. That means when you just do the crop rotation and keep doing, for example, if you are cultivating ginger for one period and next you can cultivate something else, and alternatively you can again start cultivating ginger, that is called as your crop rotation. So planning a crop cultivation, you can regularly supply the drug throughout the year. Okay, and these industries which are depending on the uh, getting the supply of these crude drugs will not face any problem in shortage of the raw material. Okay, and the next advantage is the cultivation of medicinal and aromatic plants. It also leads to industrialization. As I already told you, the cultivation of coffee and cocoa plant in Kerala, usually it has given rise to the formation of more number of your cottage and small scale industries. And then in again, in if you see West Bengal, there the cultivation of cinchona, which has been lead to the alkaloid factory near Darjeeling. And again, already we have seen in the cultivation collection of opium at Ghaziabad, the opium processing factory is there. So that is completely owned by the government. And this is again what happens, the industrialization, uh, there is a possibility where more number of employment is because of the cultivation of the crude drugs. And the very last advantage of this cultivation method is cultivation permits the application of modern technological aspects. That means the genetic engineering methods like the mutation or polyploidy or hybridization or any other genetic engineering method, so that the plants with the desired characters can be brought about. So these are all the various advantages of this cultivation of fruit drug. Okay, and next we'll see about the methods of propagation. What do you mean by propagation? Propagation is nothing but raising up of the plant. Okay, so in this, when you say majorly, the plants can be raised by two methods. One is called as your sexual method, and another one is your asexual method. So what do you mean by that sexual method of propagation? It is otherwise called as seed propagation. The plants are raised from the seeds, and these plants are called as your seedlings. Okay, so what are the advantages of this sexual method of propagation or raising the plant from the seeds? When you see one by one, the seedlings raised from the seeds are long lived, and when they are bearing, it bears more heavily. Okay, and these plants are much sturdy also. And these seedlings, which are raised from the seeds, are comparatively cheaper, and it is very much easy to raise the plants. And also the other advantage what you can see from this is it is responsible for the production of some chants. What are the chants? That is seedlings with the high superior qualities, and uh, with the superior quality, they will play an important role in the society. The examples may be in the production of orange or papaya cultivation. This plays an important role. And next one, the in case of the vegetative methods, uh, that is, if you can see some of the examples of the drugs where you cannot develop the plant by the vegetative methods, that is the asexual method of propagation. So in such cases, the raising of plant by sexual method is the only propagation or only method of choice. Okay, and these are all the advantages of the raising of plants by sexual method. That is, the seedlings are long lived, they are more heavily bear the fruits, and they are sturdier, and they are cheap and easy to raise, and they can develop some of the important characters. And also, this is the method of choice for some kind of plants which can be raised only by sexual method of propagation. And the disadvantage, you see, seedlings, they are not uniform in the growth, and the yielding capacity is uh, sometimes less when it is compared with the grafted one, or plants which are raised by a vegetative method. And these plants, they require more time to bear the fruits when it is compared with the grafted plants. And again, the cost of harvesting and spraying of the pesticides, the other uh, practices which are done during the cultivation, is also more than the grafted trees. And this one, in the plants which are raised from the seeds, sometimes the modifying influences are not possible. That is nothing but the root stocks on cyan, as you do it in our vegetative propagation. That is not possible in the sexual method of propagating the plants. And when you are raising the seeds from the plant, there are some criteria you have to look into account. So what are those criteria? Whenever you are selecting a seed, it should be of good quality, it should have high germination rate, it should be free from any diseases or insects, and also it should be free from other kind of seeds and extraneous material. And the germination capacity or the germination rate is slow, you can improve the germination rate by some of the methods. So how to study the germination rate? It is usually studied by rolled towel test or excised embryo test. That is nothing but, what do you mean by that rolled towel test? Usually you do sprouting in your house, right? In our your mom, if you see, they will be taking some of the grains and they just soak that in the water, and after 12 hours or 10 hours they'll be removing the water, and in a packed or airtight container or a towel rolled tightly, where the sprouting the next day or the next 10 to 12 hours, if you see, there will be a very nice and beautiful sprouts would have been coming out of it. Okay, so by that way the rolled towel test is done, and apart from that the excised embryo test is also done. So nothing to go more detail into this. If you just name it, it is more than sufficient. Okay. And next one, so you have found out the germination rate, and you found that the germination rate is long. That means it takes more time to come out as a sprout or come out as a seedling. So in that condition, what you are going to do? How you are going to increase the germination rate? That means what you can do is, sometimes why this germination rate is low, already when we have seen about the individual drugs, I have told you, maybe the seed coat may be very hard and strong. So in that condition, you have to loosen that seed coat. How you will lose the seed coat? Just subjecting it, or just do this scarification over the testa, nothing but your seed coat. So if you just do this scarification, what happens? The external layer, it gets disturbed, and that then what happens? It makes the permeability of water and gases to go into the endosperm. Okay, and then what happens over there? The germination can be made faster. Okay, and in this condition, if the seeds are not getting germinated, what you can do? You can store the seeds for little time in a cool and dry place, so that the germinating power can be maintained. And for this case, you are not supposed to store the seeds for a longer time. Why? Because if you store the seeds for longer time, the again germination power may be lost. Okay, the storage time should not be too short or too long. Okay, and next, how you will improve or increase the germination? Before the germination, you can do some of the treatments. As I already told you, scarification, it is just a physical treatment. Okay, you are just disturbing the outer layer so that the entry of the moisture and gases are made free, so that the internal things are getting damped or moistened, and they start germinating. And apart from that, what else you can do? You can also treat it with the plant growth hormones or regulators like gibberellins, cytokinins, or ethylene, and so that they can induce the germination. And apart from that, you can also use some of the chemicals. You can, it is given over here, you can see in the screen, that is thiourea or potassium nitrate or sodium hypochlorite. Okay, so here what happens? The dormant seeds are can be stimulated to give out the germination spore fast by treating with GA3, that is nothing but your gibberellic acid. And apart from that, the chemical potassium nitrate, if you just soak the tree seeds overnight with potassium nitrate solution, it will also fasten the germination rate. And apart from that, the thiourea, it is again a chemical. If the seeds are not germinating in the dark or at high temperatures, if you treat it with thiourea, the seeds will germinate faster. Okay, so that is the method how you will increase the germination. And next, what are the methods of sowing the seeds? When I was talking to you about the pharmacognosy of individual drugs, in the cultivation collection of the drug, I would have told you it is the propagation is done by broadcasting method or dibbling method. Especially when I spoken to you about the fennel, I would have spoken to you about the dibbling method. Okay, or the isopgol or the linseed. During that time, I would have told you about the broadcasting method. So what do you mean by that broadcasting method? That means if the seeds are extremely smaller in size, they are propagated by broadcasting method. Just that is, the seeds are taken, it is just spread or scattered freely in the well prepared soil. Okay, the examples are isopgol, linseed, or sisal. And there are some of the plants which are raised by the dibbling methods. Dibbling methods are nothing but, that is, if the seeds are having the average size or of the weight of the seeds is little more, then the propagation can be done by dibbling method. That is nothing but some few seeds. The number of seeds can be put into a single hole, and we have to maintain a different distance between the two holes. Okay, the number of seeds putting in each hole may vary from 3 to 5 based on their individual characters. What are those characters? That means vitality. Vitality is nothing but, that is what happens, that is how to which extent the seed is active. Okay, and again it also based upon the sex of the plants needed for the purpose and size of the plant coming out of the seeds. And here in case of fennel, around 4 to 5 seeds can be put into the same hole, and that can be done. And in between the two holes, the distance has to be maintained. And in case of the castor seeds, only 2 to 3 seeds can be put in each hole. And when you come to papaya, as I already told you, the plants are unisexual in nature. That means the female plants will bear the fruits, whereas the male plants will not bear the fruits. So how you have to do, you should take a little more number of seeds and put into the hole. And once the plant has come into a seedling, you should confirm what kind of sex it is. And if you see that it is a female and a healthy plant it is, you can allow it to grow, whereas the other male or other unhealthy plants can be removed from it. Okay. And the other method, which is called as your miscellaneous method, many a times what do, the seeds are sown in the nursery grounds. For example, if you take your paddy or the ragi, wheat and all, the nursery beds are prepared well, and in that the seeds are sown. Once the seedlings attain certain age or certain height, and these seedlings are taken, and then it is transplanted into the farms, and then the cultivation can be maintained. In our I have just given you an example for paddy and ragi. That is why it means you might have seen that thing. And usually in case of our crude drugs, the example for this is cinchona, cardamom, clove, digitalis, capsicum, etc. Okay. And what are the special treatments you will do to increase the germination? As I already told you, there are some things you can treat it with the chemicals, or you can do the scarification, or you can treat it with the uh, plant growth hormones like your auxins, gibberellins, etc. So here, in case of castor seeds, you can soak it in water for a day. And the henbane seeds, that is nothing but the solanaceous drug which produces the propanalkaloid, are soaked in sulfuric acid. And in case of your Indian senna, already we have seen that in the cultivation collection of senna, the seeds are grinded along with the sand in grindstone grinders, and are pounded with the coarse sand. Then it is sown in the ground. And then you can use the plant growth regulators or hormones to initiate the germination. Okay. So that is about the sexual method of propagation. And next comes the asexual method of propagation. That is nothing but the vegetative propagation. Here, the plants are raised from the vegetative part of the plant. That is maybe the different parts of the plant you might be knowing also. So like the stem, root, or rhizome, or whatever it is. Okay. And next, when you just see the advantages of this vegetative propagation, uh, you cannot see any variation between the plant grown or from which plant it has been grown out. Okay. And usually the growth is uniform in nature, and yielding capacity is also uniform. And in the fruiting trees, usually the giving out of fruits are also uniform in nature, and its quality is also uniform, so that there is no problem in harvesting and also during the marketing. Okay. And in this asexual method of propagation, the seedless varieties of fruits can be propagated. Examples may be your grapes, pomegranates, lemon, etc. And in this vegetative method, the plants will bear faster. When you just take the bearing of fruits in the seed propagation method, especially the clove or the other kind of fruits, it will take a minimum 5 years to mm, get the bearings, whereas here the bearing is very faster. Okay. And by budding and grafting, what you can do, it is one of the major advantage, that is the disease free plants or disease resistant plants can be propagated or can be brought about. And again, the modifying influences, that is your hybridization and other engineering technique, genetic engineering techniques can be made easier in this asexual method of propagation. And again, in this, what you can do when you are doing these experimental conditions, if you find that the offspring what you are going to get is of inferior quality or unsuitable variety, that also can be manipulated in this vegetative propagation. Okay. And The disadvantages, if you see, it is not that much vigorous in the growth and are not long lived, and there is no new varieties can be evolved by this method. And this is the disadvantages. And now when you see what are the different types of this asexual method of propagation or vegetative method of propagation, usually there are three types. One is the natural method of vegetative propagation. Second one is your artificial method of vegetative propagation. And last one is your aseptic method of uh, vegetative propagation, that is nothing but your micro propagation, that is which in turn your tissue culture. And when you see the natural methods, that is by using some parts of the plant in the well prepared soil, the new plants are raised out. So here I had listed out the examples, that is bulbs. Examples are squill and garlic, calm, colchicum and saffron. Tubers, potato, well known to you, aconite and jalap. And rhizomes, ginger and turmeric. Runners, peppermint. Suckers, mint, pineapple or chrysanthemum or banana. Offsets, aloes or valerian. Stolons, arrowroot or licorice. So these are the various or the different parts of the plant by which you can raise a new plant by sowing them or placing them in the prepared soil. Okay. And next one is the artificial method of your vegetative propagation. Here the plantlets or seedlings can be produced by vegetative part by using some techniques or by using some process, that is by cuttings. Usually you will be doing the when you are just you want to get rose plants, we will be doing that. So here they have listed I have listed out the stem cutting, softwood cutting or semi hardwood. So no need to write that much, very few example you can give, that is more than sufficient. And next one is the layering. Layering is nothing but just taking a part of the plant with the apical bud and it is layered on the ground. Usually sugar cane and all those things they'll be doing, right? So likewise here I have given the different types of layering along with the examples. You can give some of the examples what you can easily able to remember. And next one is your grafting. This grafting is nothing but just in this nothing but the root of a plant will be different and the uh, stalk of the plant will be different. So you are going to graft the two different plants to get a new offspring with a good and the traits, uh, the in the offspring, whatever the characters what it is going to produce is a beneficial one. Okay. So here again the whip grafting or tongue grafting, side grafting or approach grafting or stone grafting. So here along with the examples it is given that you can note down and you can reproduce it. And the last method of your asexual method of propagation is aseptic methods of micro propagation. Otherwise, you can call it as a tissue culture. What you're going to do in this method, like the natural surroundings, you are going to provide all the conditions along with your temperature, chemical, nutrient, everything in an artificial medium, and you are going to develop the whole plant from a single cell. Okay. This single cell is isolated from a particular part of the plant. Okay. That means what you're going to do over here, a single isolated cell from a part of a plant is allowed to grow in a artificial medium under aseptic conditions to develop into a intact plant. Okay. That is called as your plant tissue culture. Clear? And what are the different types of your tissue culture methods? That is nothing but the single cell culture or callus culture, seed culture, embryo culture, root tip culture, shoot tip culture, pollen grain culture, all these things. That means what? That these are all parts of the plant, right? That means nothing but your seed or embryo or root tip or shoot tip or pollen grain. from any of these part of the plant, a single cell can be isolated, and that is allowed to develop on an medium where the aseptic condition is maintained, and this single cell is allowed to grow and as a intact whole plant. That is called as your micro propagation. Once the intact whole plant has come, then it has been transplanted into the open grounds with all kinds of precautions, and then it can be developed. Okay, so in this tissue culture method, either you can develop a plant from a single cell, or otherwise you can produce the secondary metabolites which are produced in that particular plant, that can be isolated and used in the pharmaceutical field. Okay, so that is about the three different methods of your asexual method of production. Okay, so here this is the introduction to the cultivation collection of your drugs. Here we will stop it, and in the next class we will continue about the other. In this particular chapter you are going to read about what do you mean by cultivation with its advantages and the methods of propagation, factors affecting the cultivation, and also you have to study about the collection, drying, and then the storage of the crude drugs. Okay, all these things you have to read under this particular topic. Okay, now we will stop. Yes, are you able to follow?
Yes, ma'am.
Hello,
Yes, ma'am.
can you
Yes, ma'am. We're
Yes,
following.
ma'am.
okay?
Yes, ma'am.
Have any doubt? Is it okay to you?
No, ma'am. No, ma'am.
Yes.
No, ma'am.
No. Okay, fine. Shall we stop the class?
Yes, ma'am.
Yes, ma'am.
Yes.
Yes, ma'am.
Yes. Yes.
Ma'am.
Are you
No,
in
ma'am.
class? Hello.
Yes.
In the class?
Yes, ma'am.
Genius.